So my talk is not about sequestering carbon. Talk is about, as in a way the previous speaker said, it's not sequestration per se, it is actually carbon cycling. Because microbial carbon turnover is one of the key pathways for sequestration. So if you understand microbial carbon cycling, what drives it, how to manage it, how to improve it, then you are on the path to sequestration. If you go to east, you go to Sydney. If you go to west, you go to Perth. You don't want to go to west and go to Sydney. So regenerative agriculture, in a way, it's a philosophy. It's not a single term like stubble, no-till stubble, incorporated stubble. So there are a lot of definitions for regenerative agriculture. There's one definition that came up by a committee headed by CSRO Agriculture and Food Chief. And that's how it goes. Regenerative agriculture is a form of farming in which explicit attention is paid to the state and trajectory of natural capital base. And the natural capital base is soil, water, biodiversity. And underlying farm production and acknowledgement of that there are non-farm stakeholders interested in the responsible management. So it's a mouthful of a definition, trying to appease everybody that are interested in all aspects of maintaining that natural capital base. But one of the challenges of regenerative agriculture is that demonstrating that connection between soil health, again another loose general term, to productivity and productivity footprint. That's been lacking in a number of places, not only Europe, USA, and Australia. So we were involved with one of these projects, but one of these landscape SA people, and my component was to look at that collecting evidence in the low rainfall regions on farms that meet on the criteria for regenerative agriculture. I don't use the word there, actually regenerative agriculture, farms that meet the criteria to be considered as regenerative agriculture. So soil biology is again another loose term. Are you healthy? Tell me, how many of you think you are healthy? Raise your hands. See, it depends upon what is health. So much of the time, especially soil biological health is a broad term. You can measure with one measurement, but it may not relate to the whole of it. So we have been using what is called functional biology, because biota perform different functions. So it is that functions that we are interested in: accumulating carbon, providing nutrients, controlling diseases. These are the functions that agriculture people want. Doesn't matter if the farm is suitable for a road, but you want what it is doing it for. So we use this functional soil biology because the biological capabilities depend upon what types of biota are there in the soil. So first you want to know what type of biota are there. You also want to know how many are there, how active they are, and then of course what they are doing. So based on this philosophy, a number of measurements can be identified. So there is no single measure that will tell you your soil is biologically healthy or not. Because biota are diverse, the functions they mediate are diverse. So we use this approach mainly to determine the status of soil biological capacity on farmer fields, not in experimental plots, across low rainfall regions of South Australia and extended a bit of Victoria. So as I said before, even in the low rainfall sandy mallee soils, biota are diverse in terms of their size, from microbes to, you don't have many earthworms, but you do have termites. So, and the biota, all these biota are there, but they are active at certain times for certain aspects of it. But they are performing variety of these functions. Help plants withstand all of these things. You noticed I didn't put in carbon sequestration, because if you have a healthy plant, producing plant, it will lead to maintaining carbon and in situations increase carbon. So one of the main factors in this part of the world is availability of carbon. I'm taking out what some people told me, that the gods controlling things, that is moisture and temperature in this part. You can't control rainfall. But it is the availability of carbon, of course, some physical and chemical constraints. So it's availability of carbon that helps maintain and improve soil biology. So that's one of the principles that we use to but in this biota, even though there are many of these things, like from earthworms that to these are nematodes and some protozoans, 90%, if you take a weight of biota in a soil, 90% weight is actually by these microbes that you can't see, bacteria, fungi, and those things. And they are the ones primary drivers of all of these processes. So nowadays, everybody can do, many of you would have heard DNA sequencing. So you don't measure microbes anymore by growing on a plate in a lab. You extract DNA and sequence them. So if you look at that way, you can actually get what is called, a family tree of one group of microbes that fix nitrogen. So it's like you can make a family tree for yourself in this. Scientists make this type of these things, pictures. So in a gram of soil, in a mallee, there are 10 to the 9 bacteria per gram of soil. And if you look at the genes they hold, there are 10 to the 8 genes. You may say why there are less genes or microbes. That's because one type of gene can be in 10,000 species. So therefore less number of genes than microbes itself. So even in the mallee, you do have, but the difference is that in the low rainfall, low organic matter systems, their activity may be for a short period of time only. And they are prone a lot more to the effects of those factors I said. So with this base, we said, okay, let's look at what are considered regen ag practices. Once again, you can't see all of these. These are all various management practices: tillage, crop rotation, all of these. And there are a number of ways that you consider regen ag practices. So what we did is, this is too complex. So we tried to find the management practices that in the low rainfall regions that are followed into categories that are relevant, considered as the regen practices. So these came up with these 7 different types of management practices. This we can easily relate to. So tillage, stubble retention, crop diversity, cover crops or a ground cover, chemical and fertilizer applications, and we used grazing because if you have a sheep in a farm, the grazing is one of the management practice. Then we try to come up some sort of a quantitative categorization. How can you say my farm is regen ag, the other farm is not, if you follow some, not other of these practices, and you follow at different levels? So in order to characterize the farms, we gave a sort of a rating, 0 to 2. 0 means it is closer to the regen ag that generally considered. We also included in this case, because the conservation agriculture practices that have been followed for the last 25 years in this part of the world and other parts of the world also have required fertilizers and pesticides, herbicides for weed control, fertilizers for growth, because you have a low organic matter soils. So we included that as part of it. So after doing that, we went to, I didn't go at all because it was COVID, so I stayed in the lab. Other people went, and we identified 35 fields across low rainfall mallee. And then we asked farmers, how many of those farming practices, we selected 7 of them that implemented to certain level. And how many of them had growing crops, not just pastures, for the last 3 years. So if they met those 2, we collected 35 fields, and we followed a scientific procedure to analyze. And we analyze all those various parameters, 8 or 9, 10 of them. We use the parameters to measure or quantify what is the total size of the microorganisms, that is microbial biomass. How actively they are actually, I would say, eating carbon, utilizing carbon. Are they able to utilize diverse type of carbon? And are they mineralizing nitrogen and performing processes that are involved in release of phosphorus, sulfur, nutrients? So these are the 4 or 5 categories of biological processes or microbes we measured in order to obtain what is the status of soil biological capacity in these 35 farms. And another thing, most of the times microbial properties, if you ask any lab, they will say, if you have an experimental plot, you can measure microbes easily, because the spatial variability of microbes is quite large compared to the general soil physical properties, sand, silt, clay type. Because microbial populations amount and activity depends upon not only the soil carbon, soil habitat, but water holding capacity and everything else. So it is considered that measuring biology on a farm scale reliably, reproducibly is difficult. So we designed a sampling protocol. The sampling overcome the spatial variability aspect of it. So in a way, we were testing if the methodology available to measure soil microbiology, biology that is used in research can be applied to the farm scale. So then you can see these are 35 fields, and that is the amount of microbial biomass. So this actually, that is where that is 400 kilograms per hectare of microbial biomass. So that much of microbes you have in the top 10 centimeters. Each of the bar, which is very difficult for you to see anyhow, has a small line. That is the variability. So the protocol that is used produced a reliable estimate of total microbial populations on a farm, and our sampling area is 100 meters by 150 meters, or 150 by 150 meters. So in that area, we were able to acquire amount of total microbial biomass. I'm not going to say which farm and what later. The reason we did that is, it is the microbial populations, that's microbial biomass, that is the engine that drives the decomposition of organic matter, soil organic matter, crop residue, leading to, it doesn't matter which microbe is there in the low rainfall regions. If you have microbes, they will be performing, because 90% of microbes in most soils are what is called heterotrophic. They need carbon for energy to grow. So if you increase total populations of microbes, automatically you are increasing engine size. I don't need to tell you about the importance of engine size to you. So another way is, it's very easy to say this farm is better than that farm because one is higher than that. But the size of the engine depends upon the size of the energy source. So if your soil has a 3% organic carbon, you could still have higher biomass than a 1% organic carbon. But you can't compare that 3% organic carbon soil to 1% organic carbon, say you have low microbes. It is a proportion of organic matter that is in microbes that is important. So therefore, there is a measure called, the other measure, before I go to that, is nitrogen mineralization potential, ability of microbes. Again, we were able to obtain reliable measures, reproducible measures. I will come back to that. So you can calculate what is called microbial coefficient, that is amount of microbial biomass per unit organic carbon. So that's one way to compare farms with different soil texture, different organic matter, different rotations on an equal footing. So if you look at that, it is known that 5% organic carbon as a microbial biomass, there is a line there, you just have to believe me there, then that is considered that that amount of microbes can maintain the biology for that soil to perform all of these various functions. So below 2% is considered as the soil requires significant changes to improve. But you notice most of the soils that we measured are in between, not many are above. So in these soils that we measured, that the amount of microbial biomass is there to sufficient to perform functions at that level. But if you want to improve them, then you need to increase microbial biomass or increase total amount of microbial populations. One of the things is that because microbial populations, abundance, and the activity depends upon availability of moisture and all of this stuff, seasonally it changes. If you go in the summer on the same field, if you come today, you have a 1 ton per hectare, but in crop, it will be higher. So if you want to compare different systems, if they have a sufficient microbial biomass, you need to measure in crop and out crop to decide. So when we did that and looked at see if these farms differ in terms of amount of microbial biomass through the season. And you notice most of the farms, the trends are quite similar. There are some that are different. And that depends upon what management practice they had. So once again, using this approach, you were able to categorize or at least quantify the biological capacity of soil. And that is one of it. The other one is the ability of the microbes to use carbon sources. As I said before, most microbes are heterotrophic. They need energy to use. And not all microbes can use all types of carbon. Sugars, carbohydrates, proteins, and various things. So you can actually estimate the activity capability of microbes using a laboratory assay. And the results from that can be interpreted two different ways. One, you can calculate what is called community diversity, metabolic diversity, ability to use different types of substrates. The other one is if you're comparing different fields, you can do this type of a fancy analyses, and you can see this farm is quite different to that farm. So you can actually compare, if you're looking at where my farm lies across the Mali environment, you can do those comparisons. And from the different 35 fields we used, and you can see there are quite distinct differences between. So what we also did is that using this data, we tried to see what is actually driving this diversity in the different fields. So as I said before, it is the availability of energy source that drives the activity of microbes, depending upon the type of energy source, amount of energy source. And lo and behold, we had a measure of what's called active carbon, that is microbially available carbon. And once again, you need to take my word, there is a linear line there, that this metabolic diversity is actually related to the amount of available carbon. So maintaining microbially available carbon is a driving force for the amount of microbes and their ability to generally just to grow and be active. So that's one way. So we use similar approach. In this is hopefully you can see some colorimetric things. And that is about those two things I talked about. So we looked at the diversity of microbes and all the microbes involved in nitrogen mineralization, carbon cycling, phosphorus release, sulfur mineralization, all of these various things. So we were able to compare these 8 fields. Yellow means low, green means high. So using this functional approach, you can quantitatively categorize fields in order to actually understand what management practice is driving them. Once again, it becomes too complex. It takes for people like Gupta or others to explain that. What can a farmer do if you send a soil somewhere? So there is a way that you can actually combine the various properties in order to come up with an index for comparison. In this case, we call this as a multifunctionality index. It's just a fancy term that can says that it can do everything type of thing. So if it is below line, that means it is low multifunctionality index. If it is above the line, that is high multifunctionality index. So if you want that way, I'm not saying why, this paddock biology is more healthier than that paddock. So then we went and looked at what are the various management practices that these 35 farms followed. In this case, there are 7 of them. So number 1, the one that is really lowest. And what we found out is that farm and that area was under drought 3 out of 5 years. So there's not much carbon going in. So therefore, with time, the soil has lost its biological capacity. Then the second set are the ones that are moderate way. All of these 3 farms, although they had a no-till and rotational everything, they all had grazing as a major farm practice. Grazing in the sense, available carbon through crop residues or plants went through the grazing animal before it went back to the soil, available to microbes. And the ones that are on the other side are the ones where they didn't have a grazing and they had a stubble retention, no-till, conservation agriculture practices. So out of the 7 characters, and these farms did not differ much in terms of a herbicides or pesticides. They all use some level of herbicide use. There was nobody fully, like no chemical use like that. So we really could not evaluate effect of chemicals on it. We did a similar thing. Of course, this is South Australian one. In a Victorian one also we did. Again, just to sum up, all of these things is, all the ones that are below are the ones that had some level of a removal of carbon, either through grazing or hair removal. So how to interpret this? Let me see what is next. Yeah. So we did that both in the summer and in crop. And you can see most of them are, the one above is above, below is below. So in both times, the soils in these farms have a similar soil health status. So why is that? Why grazing or a removal of carbon is doing this? As we heard once before, to maintain biology with the rainfall that you receive in this part of the region, you would require a minimum of 1 ton of carbon going back in to the soil for microbes to be available. So 1 ton of carbon is 2.5 tons of crop residues. If you do not have a 2.5 tons of crop residues, approximately every year, that is going back into the soil, roots or above ground, whatever, then your microbes are always using the carbon that is in the soil. Whenever there is a nice warm temperatures and moisture, microbes use the carbon. So it is that reason why we heard in the previous speakers, increasing soil carbon in the area 600 below rainfall is tough. It's not impossible. If you manage things, it can happen with time. But if you follow these threshold values that is required to maintain biology, so it doesn't matter what your regenerative ag practice is, what your management practice is, if that practice does not allow for the amount of carbon to sustain microbes to maintain the carbon, because for each 100 units of carbon microbes use in this part of the world, 75 units is respired as carbon dioxide. Even under best conditions, 60 units is respired, like we are respiring. So only 40 units is kept inside. That's under best scenario. So therefore, agriculture practices that can maintain that carbon requirement can help maintain soil biology and ultimately carbon turnover, ultimately leading to sequestration. So another one is that, let's say your biology has reached certain level, organic carbon reached certain level. When microbes go through uniform conditions, they perform one way. When the microbes are exposed to various stress events, dry, hot weather, nice wet weather, dry, hot weather, these are with a chemical application, low carbon source because of drought and a good crop season. These are all various stress events. It is the ability of microbes to recover after a stress event is another parameter that you can use whether soil biology is healthy or not. So just pictorially to show that, so if you have in your soil, microbes going at that level, but when it is exposed to the stress, if it goes down. But can it recover? How well it can recover? So that's called resistance, and that is resilience. So not having certain level of biology is sufficient, but also whatever you have has to be resilient in order to maintain that. Like we heard before, once you reach a carbon that level, then you can't get any benefit. You can't benefit in a carbon per se, but you can benefit in maintaining that carbon. So that is important, because as soon as my carbon has reached this level, it can't go up anymore, I can do what I like, then you are going down that way, whether it is regenerative or not. So we measured the resilience of these soils along with the capacity. So we measured this microbial biomass, and we don't need to go through that because you really can't see much. Then we calculated the resilience index. Once again, the same format that you saw before. The soils we tested are all sands and sandy loams, mostly sands. In sandy soils, it's too bad you can't see the pictures, most soils are what we can call naked, because microbes in soil are not just as microbes in a flask in a lab. Microbes in soil are in protected environment, because microbes produce stuff that actually lead to the clays and silts attached to them, and then they are covered. But in sandy soils, there are no clays, so the microbes cannot build their own houses, micro houses. This is like a 0.5 millimeter aggregate. Because of that, the resistance of microbes when you expose to stress is very limited. That's the reason all these blue lines are resistance. You notice the amount of microbes when you expose to stress is very less. So all the microbes have to build up again as soon as the stress event is gone. And in some systems, yes, even in these environments, there are some soils that they all can recover. Even here they are recovering, but not to the level higher again. So all this means that in order to maintain biology and in these environments and in these soils, you need to provide those microenvironments where microbes can survive, grow, and be active. Since you don't have clays, in this case, microbes are associated with decomposing residues, roots and decomposing residues. So any management practice that provides these things, whatever it is, that can be considered in these environments as regenerative. Otherwise, it is not. The reason why grazing didn't do good is because grazing is removing that carbon source. Those not only energy, nutrient source, but also the habitat for them. So we can go and list all of these things and where these farms lie, but as I already said, it is like that. So at present, there is information. This is for conservation agriculture practices. All of these 7 categories I talked about, and there is information available so that you can apply that in these environments. And then you can see, yeah, 5 of 6 that I'm doing it, mine is regenerative. The 7th one I can't. That's because, for example, you have a weeds, summer weeds, and you have to take it out for growing a crop. Then you cannot do that if you are a growing crop. So that's how you can adapt the regenerative agriculture philosophy to the low rainfall, low organic matter soils. Thank you.